It's my pleasure this afternoon to perform a very happy task, and that is this is the presentation of the CIBMTR Distinguished Service Award. This award was initiated in 2010 to recognize the contributions of an individual who'd worked with dedication and in partnership with the CIBMTR to promote stem cell transplant research in developing countries. The award is designed to honor persons who, despite unique challenges, have established outstanding programs that provide state-of-the-art clinical care and advance the field. In collaboration with the CIBMTR Executive Committee, the International Studies Working Committee selected Dr. Santiago Pavlovsky as the 2011 recipient of this Distinguished Service Award. However, I have to say that it is with great sadness that we present this award posthumously to Dr. Pavlovsky, who passed away on September 19th of 2010. During the 1990s, Dr. Pavlovsky was instrumental in establishing a committed group of South American centers as participants in the Autologous Blood and Marrow Transplant Registry, one of the forerunners of the CIBMTR. The centers continue now their active contribution to the CIBMTR research programs. Dr. Pavlovsky's professionalism and dedication to his patients and to clinical research were outstanding throughout his life. For many years, he was the medical director of Fundalú at the Angelica Ocampo Hospital and Clinical Research Center in Buenos Aires, Argentina. Fundalú is a non-profit organization formed in 1956, almost 60 years ago, to serve the community through research, information, diagnosis, and treatment of hematologic malignancies. Dr. Pavlovsky was a member of numerous scientific societies in Europe, Latin America, and in the US, and was named Doctor of the Year for the year 2000 by the National Ministry of Health in Buenos Aires. He was on the editorial board of many journals and published over 120 articles in international peer-reviewed journals. While we are sad that we were not able to give this award to Dr. Pavlovsky in person, we are very fortunate that his daughter, Dr. Astrid Pavlovsky, a hematologist herself who worked with her father at Fundalú for many years, is here today to receive this award. And Astrid, I was very touched to hear that one of your father's major concerns in the last weeks of his life were to ensure that the data was in good order and that it continued to be transmitted to CIBMTR. So if you will come up, it would be my pleasure to present you with this award.
Thank you very much to everyone who has made this possible, not only giving the prize to my father, but also for bringing me here and having the honor to receive it. My father was very passionate in living his life, and I am sure hematology was the greatest passion he had and the one he was mostly recognized for. He made an enormous effort, and an effort he enjoyed, to bring Argentinian hematology to international standards. And that is why many years ago he was very interested in starting to collaborate with the CIBMTR. He was very serious about this, and as it was mentioned, this was one of his last concerns. We even had a meeting a couple of weeks before he died about keeping everything updated. He wanted to do everything the best possible way, and that is why he decided to be part of this international group. My father was a very hard worker, and he was a good mentor and a good colleague to everyone working in Fundaleu. So now we have the responsibility and the honor to keep up his efforts, and we are assuming with all my colleagues in Fundaleu to still be part of this international organization. And he also enjoyed life a lot in a very intense way. And one thing he always mentioned me is that one of the good things hematology had given him was the whole lot of hematological international friends he had all over the world. And I am sure that if he would be here today, he would be introducing me to all his friends, many of you sitting there. So I want to thank all of you on his behalf, and I want to thank all of you for the hundreds of mails we received after his death. And just tell everyone that back in Fundaleo in Argentina, there's a still hardworking group and friends you can count on. Thank you.
It is now my pleasure to move on to the next item on our calendar this evening, and that is, again, another pleasure to introduce to you the recipient of the Mortimer Burton Award, who will give us his presentation tonight. It is my pleasure that Armand Keating has agreed to give this award. Dr. Keating is a professor of medicine, the director of the Division of Hematology, the Epstein Chair in Cell Therapy and Transplantation, and a professor in the Institute of Biomaterials and Biomedical Engineering at the University of Toronto in Toronto, Canada. He's also the director of the Cell Therapy Program, the Orsino Facility for Cell Therapy, and the Orsino Cell Therapy Translational Research Laboratory. You're a busy man, Armand. At the Princess Margaret Hospital and the University Health Network. Armand obtained his MD degree from the University of Ottawa and completed residencies in internal medicine and hematology at the University of Toronto and a research fellowship in oncology and bone marrow transplantation at the University of Washington and the Fred Hutch Cancer Center in Seattle. He was a cancer research scientist at the National Cancer Institute of Canada for 10 years, and upon his return to Toronto, established the largest stem cell transplant program in Canada. He was the Chief of Medical Services for a decade and head of the Department of Medical Oncology and Hematology at the Princess Margaret Hospital and Ontario Cancer Institute. Armand is the president-elect of the American Society of Hematology and will become the president in 2012, and is a past president of ASBMT and chair of the steering committee for cell-based therapy of the NHLBI. Armand has chaired many NIH workshops and is the incoming chair of the Translational Research Committee of the International Society for Stem Cell Research. Armand is very active on numerous editorial boards of scholarly journals and is a co-editor of Bone Marrow Transplantation and an associate editor of the Biology of Blood and Marrow Transplantation. So Armand is enormously busy and does a great many things. On a personal note, I've been fortunate to spend a number of happy weeks in Park City, Utah with Armand on the ASBMT Clinical Research Training course. And there I've learned a lot from Armand. I've learned a lot about biology. I've learned a lot about bone marrow transplant. But the most useful thing I've learned is how to pick the best bottle of wine on the menu. And that's an education he can well share with all of us. So Armand, I'd like you to come up and receive this award. You have to jump. You can also jump.
Well, thank you very much indeed, Stella, for that very kind introduction, and good evening, ladies and gentlemen. I must say it's a great honor and humbling experience to be invited to deliver the Mortimer Burton Lecture. I didn't know Dr. Burton very well, but in my limited experience, I found him to be a very caring and compassionate man, and also, I think, a visionary. I should have prefaced my remarks by saying I have nothing to declare, no conflicts of interest to declare about this. But the visionary aspect of Dr. Burton, I think, can be exemplified by the data which indicate that in 1968, and he started the IBMTR and became its scientific director in 1972, there were a total of 203 patients in the registry, and of whom 3 were alive. And the visionary aspect of it is that since 1992, the IBMTR and then the CIBMTR has helped to spearhead experimental and observational research in blood and marrow transplantation under Mary Horowitz's leadership, such that there are now more than 240,000 transplant recipients from more than 400 transplant centers worldwide, coordinated by CIBMTR, and that's based on over 350,000 transplants that have been performed and over 20,000 cord blood transplants that have been conducted since the first one in 1988. And the remarkable thing is that more than 620 papers have been published on the research that's been conducted. So the achievements and the vigor of the field have been accomplished by clinician investigators working in conjunction with researchers conducting laboratory and preclinical studies, and I think they fail to do so at their peril. And what I'd like to do is to recognize a success, and that is Ernest McCullough, or Bun McCullough as he was known to many people, who died 5 weeks ago at the age of 84. And Bun, together with Jim Till, developed what could be considered a functional stem cell assay, the CFUS, colony-forming unit spleen. And you can see the bumps here, and each bump represents a progenitor cell or a stem cell that gives rise to many lineages and in fact is clonal based on this iconic graph that was published in 1963. Now, the activities in cellular therapy are reminiscent to me of the early days of blood and marrow transplantation. After all, BMT is perhaps the most mature and successful form of cell therapy and should serve as a model for the field. And I think it's useful to learn from the BMT experience when we consider grappling with this burgeoning activity in cell therapy. There are 2 areas that I'm going to allude to relatively briefly. One is regenerative medicine, and the other is cell-based anti-cancer therapy. And there are obviously going to have to be general comments that I'm going to make. There are many hundreds of clinical trials that are underway worldwide in cell therapy. We estimate that there are more than 4,000 patients, for example, who have received mesenchymal stromal cells for a wide variety of disorders. But most studies are single-arm trials in single centers with small numbers of subjects. Industry, however, after a very significant lag period, has become involved as the rationale to proceed to later phase clinical trials, phase 2 trials, but particularly phase 3 trials emerge for particular indications. One of the notable things about regenerative medicine is this extraordinary convergence of diverse fields over the last 20 years. I can't think of any other area where this really applies, and it extends from tissue engineering to developmental biology, materials science, genetics, and molecular biology. So it was in this context that if one could reduce the complexity of cell therapy, it might be feasible to use it worldwide, including in developing countries, to address enormous unmet medical needs. And to that end, Heather Greenwood from the University of Toronto convened an expert panel with representatives from the countries that are shaded in blue to rank the applications for regenerative medicine according to the criteria that you see here. So burden, a pressing health need, impact, improvement over existing therapy, feasibility that really needs to be done within the next decade. Affordability, acceptability, that's both safety, social, and legal criteria, and then indirect benefits, capacity building, economic growth, and so on. And I thought I would share this list with you because I think it's really very interesting. And although it was developed for individuals in developing countries, I think it applies equally well to the developed world as well. And here's the ranking. It's kind of an interesting ranking. Number 1 is insulin replacement and islet cell regeneration for diabetes. 2, heart muscle regeneration. Immune enhancement for infectious diseases. Tissue-engineered skin substitutes. Biocompatible blood substitutes. Interestingly, and somewhat surprisingly to me, umbilical cord banking for future cell replacement therapies was way up 6th. And then tissue-engineered cartilage, gene therapy and stem cell transplants for inherited blood disorders, nerve regeneration, and hepatocyte transplantation for chronic liver disease and liver failure, which is a huge problem, as you know, in Asia. Now, I'm not going to provide you with a list of all the indications and studies that are underway in regenerative medicine. This actually is such a list because there have been studies being conducted in all of those indications. So the question that I was pondering was, how has cell therapy fared from basic science, translational research to early phase clinical trials? And we have, I think, a pretty reasonable model in blood and marrow transplantation to compare this with. Well, the history dates back to before 2000, but that, at least in my experience, was when things really took off. And the notion in the year 2000 was that tissue regeneration was driven by transdifferentiation of donor stem progenitor cells. And you may recall the studies that were reported by Don Orlick, in which he used an experimental acute MI murine model and measured hemodynamic parameters in that model. Was one of the reasons that he chose it, because there was a good and hard and reproducible endpoint, and concluded that because the marked donor cells, these were hematopoietic cells, the marked donor cells co-expressed a cardiac marker, well, maybe I think it was 2 cardiac markers, that in fact the mechanism that was taking place was transdifferentiation. And this was supported by other studies. Now, I've just chosen one from Marcus Grompe's group, looking at a murine tyrosinemia liver failure model with the FAH enzyme, where there's a lack of this enzyme, but this defect can be corrected using KLS cells, so murine hematopoietic stem cells. The observation that Eric in Marcus's lab made was that there were a large number of FAH donor cells within the parenchyma, and that these cells, in fact, expressed hepatocyte characteristics. But around about that time, from late 2000 to early 2001, and right up to 2003 and 2004, there were opposing views. Now, I'm not going to catalog them because I don't have time to do that, but the explanation was that this was acquisition of host cell characteristics as a result of cell fusion. And in fact, Marcus's lab went on to show exactly that phenomenon. And here you can see this is a sex mismatch demonstrating a fused hematopoietic and hepatocytic cell. However, further studies indicated that the frequency of cell fusion was low, and it was unlikely to be the key explanation. In fact, in many studies, it was as low as 0.01%, and in my own hands, it was around 0.8%. So we were also interested in addressing this issue, and both in vivo and in vitro studies, again using a mouse model, we demonstrated that the donor cells, in fact, co-expressed both cardiac and stromal characteristics. So that the conclusion, in fact, was that none of the stromal characteristics were lost despite the acquisition of the characteristics of the host damaged tissue cell population. And of course, this had been missed because previous investigators didn't actually look for the presence of the original presence of the markers for the original donor cell. The corollary to those studies, and I should add that we demonstrated, in fact, that there was hemodynamic improvement in that model, was that acquisition by donor cells of these characteristics of injured host tissue cells was partial, and that it probably had little impact on the therapeutic outcome. But this, and it's interesting how scientific fashion motivates endeavors. And of course, it's understandable because there was this tremendous unmet medical need, particularly in cardiac regeneration. So there were in a relatively short time, there were over 1,000 patients accruing to over 18 trials, including 13 randomized studies that were conducted. And in a paper published 3 years ago from the Cochrane collaboration, 811 patients were studied, examining the impact of autologous bone marrow-derived cells. And what you can see here is an interesting outcome, highly significantly different. What we're doing is looking at different ways to measure ventricular function, left ventricular function. And you can see that this is highly statistically significantly different. But the clinical impact of this is rather limited. And the authors of this particular study concluded that indeed it was modest compared with conventional treatment. But the approach was safe. However, it does raise the issue of whether or not these were the right cells to study. Were these clinical trials prematurely initiated? And it was around 2006 and 2007 that the mantra changed. And it became, based on excellent data as well, became that donor cell-mediated tissue regeneration results now not from transdifferentiation, but from the release of paracrine factors. And I've just taken one example of this. It's a compelling example. I think this was published in Cell Stem Cell from Darwin Prokop's group, in which Dr. Prokop demonstrated that mesenchymal stromal cells, in fact, lodge initially and predominantly in the lung, causing microembolization. And a consequence of that is the release of an anti-inflammatory protein called TSG6, which has been known for quite some time, and in quite an elegant study was able to demonstrate with loss and gain of function that TSG6 was at least partially responsible for that improvement in cardiac function, suggesting that there was some kind of a major anti-inflammatory mechanism that was playing out here to improve tissue regeneration. And in my own lab, Victor Dayan, who was a postdoc from Uruguay, used an immune-deficient mouse model of acute MI, where we looked at the effect of mesenchymal stromal cells. And I'm going to go through this pretty quickly. What we demonstrated was that there was an overall decrease in macrophages that infiltrate the ischemic myocardium in this experimentally induced MI. But that, and the normal initial response is that the macrophages that infiltrate are pro-inflammatory, which sets up a series of reactions that results in inappropriate remodeling, eventually leading to significant scar formation and congestive failure, which is really the main cause of death in many of these people. So in the experimental animals, what Victor demonstrated was, in fact, that there was an increase of macrophages that were activated in an alternative fashion, resulting in anti-inflammatory responses. And this is further shown by looking at the ischemic tissue in the animals, demonstrating that pro-inflammatory cytokines were reduced and anti-inflammatory cytokines like interleukin-10 were increased. And that this was also associated with the reduction of apoptosis of those cardiomyocytes, so a protective effect. But in addition, he was able to show that conditioned medium could do the same thing. So you didn't need cell-cell contact to mediate that process. And furthermore, that blocking interleukin-10 could, in fact, reduce the number of anti-inflammatory macrophages, the M2 type macrophages, and reduce hemodynamic improvement. So the conclusion from that particular study is that there was a switch going from M1 to M2 macrophages that infiltrate the myocardium, which was mediated in part by the elaboration of interleukin-10 from the stromal cells, and that this was accompanied by an improvement in ventricular function. And the reason I raise this is that it immediately suggests new preclinical models that could be designed to test the utility of engineering those MSCs to express interleukin-10 and further enhance cardiac regeneration. Now, a number of these approaches, of course, are not tremendously hypothesis generating, so they may actually, and this is an issue that we've been grappling with, or the NIH has been grappling with, that preclinical studies really need to be funded in a different way, because they're not going to compete terribly favorably with hypothesis-driven questions and proposals. So the conclusion from this vignette that I've provided for you is that translational research and preclinical models invariably inform the design of clinical protocols, but clinical investigators must be mindful of preclinical developments. The preclinical work, in fact, was way ahead of the clinical, and understandably, way ahead of the clinical studies that had been conducted, particularly the randomized clinical trials that had been conducted. And so my point is that an ongoing partnership with laboratory investigators, I think, is really important. And the concern is an obvious one. The concern is that we don't want to miss a potentially effective therapy because of masking as a result of cell population heterogeneity or focusing on an ineffective cell type. Well, let me move quickly to cell-based anti-cancer therapy, and this is a huge topic, and you've been hearing about it in considerable detail, many of those talks from people in this audience. So I'm not going to dwell very much on it, except to say that the Don Thomas lecture given by Malcolm Brenner is actually going to focus on cytotoxic T cells. So I'm not going to talk about that at all, but I'm going to focus, and because I believe it's a platform, I'm going to focus on NK cell immunotherapy and the challenges that are faced by outstanding investigators like Jeff Miller, for example, in this area. It's very challenging. One of those challenges is the variable ability to generate adequate numbers of autologous or allogeneic NK cells. And the need, the perceived need, the reasonably perceived need to ensure the in vivo expansion of those adoptively transferred NK cells. And the consequence of that is that one directs the technology towards enhancing lymphodepletion with cytotoxic and other agents. And the problem there, of course, is that that is also going to enhance toxicity and, in a sense, deflect from focusing on the efficacy of this particular approach. So an alternative approach is to consider something perhaps unusual, and that is the clinical use of permanent NK cell lines. And as far as I tell, there are about 5 such lines in captivity. NK92 is the only one that, to date, and it is the most effective to date, that has been used clinically. So this line was isolated by Hans Klingemann when he was in Vancouver from a patient with LGL lymphoma leukemia, and that was in 1992. And this is a permanent neoplastic line. And up until very recently, there was no evidence that, in fact, it would form tumors in immune-deficient mice, although we have used Nodskid gamma null mice and can now demonstrate the presence of tumor formation with this line. What's characteristic about this line is that it has a lot of activating receptors, but very few KIRs. And it doesn't express CD16. So the 2 pathways that are most likely to serve cytotoxicity with the NK92 line are the perforin granzyme mechanism and the FAS-FAS ligand approach. And this is work that we have been conducting in conjunction with Ralph DaCosta at the Ontario Cancer Institute using a window chamber model. And here you can see what is called granule polarization, where the granules, in fact, coalesce or at least come together in apposition to the target cell. I believe that this approach using the window chamber model is going to be very informative because it will tell us about the kinetics of the interaction between NK cells or at least these NK cells, and the malignant targets. We've also used quantum dot labeling to look at this because I wanted to see whether or not the mechanism could actually be documented, that this was really granzyme related. And here you can see evidence of the intrusion of those granzymes and perforin into the target cell. But NK92 also appears, this is work done by Brent Williams, who's a pediatric hematologist who's doing a PhD with me, in which he was able to demonstrate that there appears to be some selective cytotoxicity of NK92 against clonogenic leukemic cells. And this can be missed if one doesn't perform the old-fashioned clonogenic methylcellulose assay. If you look at bulk cells, or if you attempt to look at immunophenotypically defined stem cells, 34 positive, 38 negative, 123 positive cells, for example, one may actually miss that as well. So the advantages of using NK cells clinically, NK cell lines clinically, is that they can be rapidly expanded to therapeutic numbers, so probably more easily than primary NK cells. The remarkable thing is that they have significantly greater cytotoxicity over normal NK cells, and since we don't want them to proliferate, the important question is their cytotoxicity affected by irradiation? And the answer is no, it's retained. And the other advantage from a cell manufacturing point of view is that these cells are homogeneous and we know their functional capabilities. And a further advantage is that relatively readily they can be modified by engineering CARs to improve tumor targeting. And we're actually now modifying NK92 to express a chimeric antigen receptor for CD123. The other advantage, of course, is that you don't have to worry about in vivo proliferation, because that's not an objective. The cells are irradiated and they're not proliferating. And that limitation can be overcome by using high numbers and several infusions. So and I'm a little reluctant to talk about this study because it's not finished, and I'm just presenting it for information rather than giving you a detailed analysis of it. It's a phase 1 trial, a classical 3x3 study with dose escalation, and it's in patients with blood cancers who have relapsed after auto transplant. And the vast majority of these patients, in fact, all of these patients in our experience, after a few months, receive palliative chemotherapy and are not actively treated. So in this study, we irradiate the cells, as I mentioned. The protocol involves day 1, day 3, and day 5 infusions on a monthly basis for up to 6 months. There is no immunosuppression or recent cytotoxics that are provided, nor is there interleukin-2, because I felt that I didn't want that to confuse the issue in terms of toxicity. And we've entered 8 patients, so there's one more patient to go. It is very well tolerated, and of importance is that there's no major effect on peripheral counts after transplant. And somewhat remarkably, there is one patient who is in a complete remission, very unexpectedly in my view, out 3 years now since infusion. So the platform is that I think it would be very reasonable to consider using this approach when disease is minimal, because innate immune cells don't require high antigen load to be effective, and the intensive therapy setting as a suitable setting, and eliminating minimal residual disease after autotransplant, in my view, is also particularly attractive. And as you all know, there are many examples of reduced relapse, but no improvement in overall survival after autotransplant for a variety of diseases, including acute myeloid leukemia. And I just draw your attention to this paper that was published in Blood last month from the EBMT, looking at autotransplants for CLL versus observation. And once again, you can see event-free survival favors autotransplant. Probability of relapse is less, but the overall survival is the same. So this is the probability of relapse. Overall survival is not different. This is an excellent setting in which to test this model and then bring those results back to the bench from the bedside. So there's been a great deal of activity that's been going on in cellular therapy, both regenerative medicine and anti-cancer cell therapy. And we first proposed to the IBMTR Advisory Committee that a cellular therapy database registry be created at that Philadelphia meeting. I'm sure that everybody remembers that meeting because of the snowstorm, and that was in 2002. And the rationale for this is that very reminiscent of the early days of transplantation. Most cell therapy studies, single-arm trials, small numbers of subjects, heterogeneity with respect to patient disease and cell protocol specific characteristics, very few randomized control trials, and importantly, and of considerable concern to me, was that there was no attempt to look at long-term effects. And of course, given that most of the studies in any one center have small numbers of subjects, detecting a relatively uncommon adverse effect was unlikely. So the parallels between transplantation and cellular therapy are really striking. So the case for the CIBMTR being involved is the following. There are many investigators who are the same in the two areas. The technologies are similar. They share an understanding of stem cell biology, and of course, the CIBMTR has leadership in data collection and analysis and an existing platform. Now, there are significant challenges, and we discussed some of those challenges in the committee meeting this afternoon. Many investigators are in fields totally unrelated to hematology or blood and marrow transplantation. Many groups work in isolation, and many of the investigators in this area don't interact with cell manufacturing facilities and scientists there who provide service for blood and marrow transplant programs. So with Mary Horowitz's strong support, we have Marcello Pasquini, the scientific director of this new committee, coming to the rescue. And this committee has now been established. Joshua Hare has now rotated off the committee. He's a cardiologist and has been replaced by, I'm sorry, I've forgotten. Can someone remind me? Mitch Cairo, thank you very much. The multi-talented Mitch Cairo, who will take on the co-chair of this. Now, we've already undertaken some studies, and there's been a survey conducted because we had no idea what studies were being conducted in cellular therapy. 115 patients have been reported. The results are as follows. Cardiovascular is leading, and this is in 2002, followed by neurological, autoimmune disease, and you can see it's based on donor type and disease indication, allogeneic and autologous. And in 2009, the number of cases of cardiovascular regenerative medicine have increased, as have neurological. Many of these, in fact, have been from Joanne Kurtzberg, looking at cord blood in patients with a variety of disorders, including cerebral palsy. So most of the centers that responded were current transplant centers, and the most common indication currently is for acute myocardial infarction using unmanipulated blood cells, and in cerebral palsy, as I mentioned, using autologous cord blood. And the mesenchymal stromal cell work was mainly for inflammatory bowel disease on an OSIRIS protocol. Now, one of the important aspects for blood and marrow transplantation is the mechanism is that we need to know a little bit more, because it's sort of fallen between the cracks somewhat, is what's happened to donor cell infusions. And so there's been a survey of this, and the data indicate that the most common indication for DCI is relapse, but some are planned, some are for mixed chimerism, for GVH, for loss of chimerism, and for viral infections. So, in conclusion, I think this is a very promising approach that's being taken, I believe. It builds on existing CIBMTR infrastructure and expertise to facilitate the development and implementation of a cell therapy database. The activity survey is helpful, but it's just the beginning, and it will help to identify the transplant centers that are actually participating in these studies. And the availability of a data repository on cell therapy for regenerative medicine and anti-cancer cell therapy, I think, is going to be an important resource for the field. And I think that Burton would be pleased with what has been accomplished in the direction that we're taking. And finally, I wanted to acknowledge the extraordinary support and intellectual curiosity, wonderful leadership, and vision that Mary Horowitz has demonstrated. It's been a real honor to collaborate with her. And I also wanted to thank Bob Gale for his rigorous but constructive skepticism for over 20 years that have kept me honest. And finally, the inspiration for my working in this area is this wonderful team that I have that crosses both the lab and the clinic. Thank you very much.
Armand is happy to answer questions if we have any. If you could come to the microphones.
The only thing is I may not remember your name.
I was wondering if I was wondering if you plan to include patients enrolled in gene therapy trials in your cell therapy registry.
It's a very good question, and in fact, Helen Heslop has done precisely that. One of the frustrations that we felt a number of years ago was that although the RAC and the FDA have data on this, the data aren't that terribly accessible, and of course there have been a number of investigators who have been working in this area from different centers. So that study is underway, so absolutely. Oh, sorry.
This is a very thought-provoking talk, and I was curious, you know, it would seem to me that this is the natural place of what we do to infuse more stem cells if patients develop organ damage from the stuff we do to them. And yet, I'm unaware of, you know, definitely trials that are looking at, you know, doing supplemental stem cell infusions for patients who develop organ toxicities from high dose of chemotherapy, which makes me wonder either we haven't done it or we've done it and it hasn't worked and nobody wants to talk about it. What have you heard?
The answer is probably all of the above. And the issue is I'm not sure that we necessarily have the most appropriate readouts. There are data to suggest that cells may in fact correct some damage. Data with respect to the use of mesenchymal stromal cells in ameliorating the actual tissue damage that is sustained after GVH, after acute GVH. So I believe that really needs to be done. But this leads me to another point which I didn't make, and that is that one of the distinctions I think we need to make is the distinction between cell therapy and cell replacement therapy. I think cell replacement therapy probably speaks to the need for using stem progenitor cells. Cell therapy, on the other hand, may not. And one may, in fact, be, particularly if the effects are based on immunosuppression and anti-inflammatory responses and so on, the focus on stem progenitor cells may be misplaced. So I think we need to keep an open mind and call it cell therapy. And maybe that's why we haven't really picked up on it.
Armand, I enjoyed your talk very much, as always. So I have a very practical question, not scientific at all. How is society going to pay for all this? So we're all, you know, the country's bankrupt, Canada's bankrupt, Europe's bankrupt.
Yeah. Well, I guess we could just print some more money, which I guess we're doing. I don't know the answer to that. It's an interesting issue, and let me give you an anecdote. It's sort of an indirect way of addressing your point, and that is that when I was in Cairo last year, or was it 2 years ago? Anyway, it was a less exciting time than it is now. What I was told was that it is much cheaper to perform a matched related transplant for patients with CML in chronic phase than to provide imatinib. So, and it goes back to my original point, which is that if we can engineer some of this, bring the cost of goods down, as they say in industry, this may in fact be just as effective and significantly cheaper than what we currently call targeted therapy now.
Thank you again, Armand.